without any further ado, please to Mr. Ahmed, the time and place is yours. Thank you.
Oh, thank you so much. Should I share my screen first?
Yes, please.
So now can you see my screen?
Not yet, sir.
Yeah,
Okay.
already.
Okay, we can see it now.
Oh, okay. So hello everyone. Firstly, Assalamu alaikum wa rahmatullah. Let's start with Bismillah. So my name is Ahmad Al Faradni. I am very happy to be with you now in this small lecture. And I should thank firstly my brother Dr. Muhtar Khair for this invitation. I am working as a lecturer at Damanhour University, Egypt. Damanhour is one of top 10 university in our country, and I am now currently working as a GCBS postdoctoral researcher at the Tokyo University of Agriculture and Technology. Uh, Hopefully, we'll start in March, inshallah. Today, I would like to talk with you about bacteriophage application in agriculture and food safety. So, let's start our conversation with about question. Do you know about what about bacteriophage means? Is there someone know bacteriophage? Do you study bacteriophage in your undergraduate study? Oh, hello,
Okay, Dr. Ahmad,
Yes.
someone is
yeah,
someone hear me?
yeah,
Yeah,
yeah.
yeah, yeah.
Uh, in our study program, we learn about the basic of microbiology. Maybe,
So you know bacteriophage, yes? Okay.
okay,
So a bacteriophage, also called a live microorganism called a virus of bacteria or viral agent that eat or kill bacteria. So bacteriophage, oh, sorry, I have some problem in presentation. Virus that infect and parasites bacteria are known as bacteriophage. So the bacteriophage name as it is means one of type of the virus that classified of virus family, and it was discovered by Frederick in the 1995, and they take its name by Felix Deherel in France 1917. So he named his this microorganism as a bacteriophage. When he sees this bacteriophage eat the bacteria, sorry. So he said that the bacteriophage in the Greek name or the Greek language means bacterial eater. So this is the bacterial eater or the virus of the bacteria can solve many problems nowadays we face due to antimicrobial resistance. As you know, there is a great epidemic nowadays called antimicrobial resistance, where bacteria can resist most of antibiotic or to be brought antimicrobial working nowadays. So even if some disease or some cases can solve by antibiotic, maybe they go to this because there is no specific antibiotic can treat such cases. Virus of bacteria or the bacteriophage can be a good solution to treat this bacteria. In the beginning, bacteriophage was used as bacteriophage therapy to treat bacterial only in animals or poultry as specific. So this is the beginning use of the bacteriophage. After that, they turned to use this bacteriophage as a human therapy. And finally, they used now to be a broad application for the bacteriophage in diverse of aspects like environment and agriculture and the food safety, animals and human therapy. So as we know, all viruses are harmful to human, like coronavirus that happened the last 3 or 4 years is an epidemic that's harmful to human. But this bacteriophage is a good virus of this virus family. There is 10 or 10 or 100x of phage more than bacteria.
Soalnya gambarnya virus.
Okay, should I complete?
Yes,
Okay.
sorry for the interruption.
Okay,
You can continue.
okay, no problem. So this virus or this good virus, let's say good virus of bacteriophage, there is found in the environment or the nature by 10x more than the bacteria. This first name, as we said, is called by Felix de Herel in 1917, is the bacterial eater or the bacteriophage. The first one used as treatment of salmonella in poultry in 1919. And the modern phage therapy now has been used to in agriculture to treat some disease cause for the plant or agriculture. Okay, the future now they wanted to make the future a phage. They wanted to make a phage bank in Asia and Africa. They wanted to make a phage bank to be a stock for bacteriophage to treat most of bacteria. Unlike the antibiotic, phage are highly specific. The bacteriophage are highly specific. There is many kind of antibiotic which work as a broad spectrum antibiotic and they work for many bacteria or many other infectious agent. But the bacteriophage can work specific for a specific strain of bacteria to overcome this bacteria and overcome the resistance in this bacteria. Sometimes when we use the antibiotic for several days or use antibiotic for several times, the antibiotic become more toxic to the human or plant or any environment or any microbiome of the animal. The microbiome is very important for human body, very important for agriculture and animals body. So when I use antibiotic for long time, this disrupts the microbiome environment. But bacteriophage on the opposite side, bacteriophage can save this microbiome because they are very specific to a target or specific to a specific strain of bacteria. This is structure of bacteriophage. The bacteriophage can consist of a head and a tail. The phage head, as we see here, this is the phage head. We can see the phage head contains the DNA material of the bacteriophage, and after that there is the tail sheath, and at the end is the tail fiber. The phage head contains the code protein and the genome or the DNA, and this genome mainly is a double-stranded DNA, which code for more enzyme and the proteins that necessary to replicate and produce new phage and make lysis of the bacteria. The tail sheath, it is about a hollow core covered with a contractile sheath. This is like a neck or like a tube for traveling the DNA from the head to enter inside the bacteria. And finally, there is a tail fiber. This tail fiber work like anchor or a fixative, like fixation of the bacteriophage on the surface of the cell membrane of the bacteria. This is the shape of bacteriophage. Mostly of us know this shape. This is by electron microscope. show us how the bacteriophage can transfer the genomic DNA from the head to transfer through the core or tail cheese to enter inside the cell membrane. There is a million of bacteriophage can enter or can fix on the surface of the cell bacteria and insert their DNA or genome inside the bacterial cell.
Iya, tugasnya aku yang ngono kuwi lho.
There is many application nowadays. There is many application for bacteria. This is photo or this illustration I produced from our publication, previous publication. So this application of bacteriophage, there is many kind of application nowadays for bacteriophage. Bacteriophage can control the fish pathogen in aquaculture. So many disease of bacteria of fish, many disease of fish now caused by bacterial agent or causative agent. So sometimes we use bacteriophage to control fish pathogen. Also in agriculture, which is our main topic today, the agriculture, most of bacteriophage can control the phytopathogen or the bacterial causes disease or disease to the plant. So pathogen can control this phytopathogen in plant or in soil. Also, we can use the bacteriophage for water treatment. As we know, most of our water sources nowadays are rich with bacterial infection. So this is very harmful to animal, to human. So this is very important to food safety. So we can treat the water intoxication or water with bacterial intoxication with bacteriophage. Also, we can use the bacteriophage to control foodborne pathogen like salmonella and listeria. This is main kind of bacteria that cause poisoning to the food in supermarket and in industry or factory of food. So we can use bacteriophage to control either the foodborne pathogen or the foodborne biofilm. Finally, nowadays, they use the bacteriophage as a biotechnological tool to detect some specific species of bacteria using the detection system of bacteriophage or the phage display system, or they sometimes can use the bacteriophage as a vaccine delivery or antibiotic delivery to enter inside the bacterial cell. This are some bacterial disease caused in agriculture. As we can see, there's a leaf blight disease on onion. We can see the disease or in the leaf part of onion, and the fire blight on apple, like this one, black rot on cabbage, like this one, black rot in cabbage, and the bacterial wilt of tomato, like this one, and the bacterial spot of peach. So there is many bacterial disease with different causative agent of bacteria can cause harmless or harmful to the plant or the agriculture environment. So sometimes we use the pesticide or antibiotic to treat this bacterial agent, but due to the microbial resistance or the bacteria resistance, we can found a problem now to use this antibiotic to treat such bacteria. So bacteriophage can use now to treat this bacterial agent in agriculture. So how do bacteriophage affect the phytopathogen or bacterial as a whole? There is two kind of cycle of bacteriophage, one called the lytic cycle and the other one called the lysogenic cycle. The lysogenic cycle is not so important in our lecture here, but what is important as a therapy or a treatment for the agriculture is the lytic cycle of bacteriophage. As you can see here is the first step. This first step of bacteriophage, the bacteriophage combined to the cell membrane of bacteria and insert the DNA inside the bacterial cell. After that, the phage DNA circulates and enters the lytic cycle of the bacterial cell, and replication start. And after that, in the step number 3, we start to make assembly of new bacteriophage and produce more than one bacteriophage inside the bacterial cell. After some time, this bacteriophage produce some protein like endolysin to disrupt the cell membrane of the bacterial cell and produce a million or billions of new bacteriophage which can invade another or other bacteria. So this is the lytic cycle of bacteriophage. This lytic cycle of bacteriophage should pass by some steps. This is the first step of bacteriophage called eclipse time or latent period. This latent period, it like this one, the manipulation and increase the number of bacteriophage inside the cell to produce more bacteriophage. After the eclipse time or the latent time of bacteriophage increased in number, there is a rise or peak in the number of phage to infect the bacterial cell. This is called extracellular phage. The extracellular phage start with disruption of the cell membrane and make lysis for the bacterial cell. This illustration summarize what about the lytic cycle? Summarize the case of lytic cycle of bacteriophage. After increase in the replication, assembly of new bacteriophage, and after that rupture for the cell membrane of bacteria and invade another bacterial cell. So there is a term called multiplicity of infection. So the question now, how many bacteriophage should infect the bacterial cell? Some answers say that one bacteriophage can kill one bacteria, but this is theoretically one bacteriophage can kill one bacterial cell. But in practical applications, this is not true. Sometimes we should measure the MOI or multiplicity of infection to determine how many bacteriophage or how many virus we need to infect the bacterial cell to kill or destroy this bacterial cell. In our experiment before, this example of lytic cycle, this one of our work in Tokyo University of Agriculture and Technology in Japan, we produce bacteriophage. We named it after that called phage 21. This is the growth curve of bacteriophage 21. This is the latent period of the bacteriophage or the time needed by the bacteriophage to increase in number or replication of the bacteriophage to increase in number. And this is axis shows the number of bacteriophage per time on the x-axis. After the increase in number of bacteriophage, there is rise or sharp increase in the number of bacteriophage, and in the same time, there is a lysis for the bacterial cell. To measure how much bacterial cell destroyed or killed by the bacteriophage, we measured something called optical density or turbidity assay. Here, we should measure the MOI or the multiplicity of infection. Sorry. We should measure the multiplicity of infection. This multiplicity of infection, we make a different dilution of bacteriophage to see how much dilution is needed to kill the bacterial cell. For example, bacteriophage MOI 1 means one bacteriophage for one bacterial cell. MOI 5 means 5 bacteriophage needed to kill bacterial cell. And the MOI 10 means 10 bacteriophage to kill one bacterial cell. So this is the control point, this control point, and this one is different dilution of bacteriophage. This different dilution of bacteriophage can used to measure the optical density. Here we found that the MOI 10 is the greatest one or highest one to destroy the bacterial cell. So if we increase the concentration of bacteriophage, we should result in highly destroyed or high killing of activity of bacteria. So if we use the bacterial infection, if we use the bacteriophage and to infect the bacteria in agriculture, we should save the quantity and the quality and the economy. I mean, if we use in this slides. If we use bacteriophage here, we can save most of our economy, and we can save more quantity and the quality of our crop or plants. So if there is a bacterial only or antibiotic resistance, we should found a decrease in the quantity and the quality of the crop or agriculture. And if we use the bacteriophage to treat this bacteria, we can result it in high quantity and high quality, which refresh the economy of our country. So our question now, what is the advantage of bacteriophage, or why use bacteriophage over antibiotic? Both of them, bacteriophage and antibiotic, can kill bacterial cell. But what is the important, or what is the advantage of bacteriophage over the antibiotic? Here is the advantage or the benefits of using bacteriophage. First one, the bacteriophage is highly specific. Highly specific means that the bacteriophage can kill only one strain of this bacteria. I means here we can save the microbiome that we speak about in the beginning of the presentation. So if we use a specific bacteriophage to specific pathogenic bacteria, here we can save the microbiome of the human or environment or animal health. This bacteriophage is safe for human body. So if we use antibiotic, we should wait for some time to the withdrawal time of the antibiotic to get out of this residue from the plant or meat or any other thing like this for human health. So this bacteriophage is safe, and there is no withdrawal time for the bacteriophage after treatment. So after treatment, we can eat the plant or meat or any other food for human consumption directly after treatment. This one is higher acceptance. Higher acceptance means some antibiotic can make easily resistance to bacteria, but most of bacteriophage slowly or low resistance can be found after using this bacteriophage to treatment. Narrow spectrum is the same as highly specific. Both of bacteriophage and the antibiotic can treat the bacteria, but the bacteriophage has a narrow spectrum to treat a specific strain of bacteria. This one, easy to control the bacteriophage. Sometimes it is advantage and sometimes it is not advantage. It is advantage and sometimes when I use the bacteriophage to treat the bacterial cell, we can control the dose as we need. But sometimes this dose can't be controlled in some cases because this bacteriophage is a live microorganism and easy to make replication and the increase in the number, which sometimes makes harmful to the human body or animal body. Affected limited to site of infection. So this bacteriophage can't be replicated unless only in the site of infection. So the bacteriophage replicate only when the host of bacteria can be found is this site. So for example, if I have problem in one site of the plant or one site of the animal, this bacteriophage only replicate in this site because the site of infection contains their host, which make replication to this bacteriophage. Genetically engineered, sometime, as we said in the first of the presentation, some application of the bacteriophage can be used to biocontrol like virus detection or vaccine delivery. So sometime we can use this bacteriophage for genetically engineering therapy. And the cost effective, the method of isolation and replication of bacteriophage inside the industry or the laboratory is easy than the antibiotic and cost effective than antibiotic. Now here, the bacteriophage in food industry. Sometimes we hear the term from farm to fork. From farm to fork means from the agriculture or animals to human consumption. means this bacteriophage therapy can used from the farm to treat the animal disease or poultry disease or agriculture disease. So this one of bacteriophage therapy can used as a pre-harvest pathogen control. We can treat the pathogen or bacterial disease before harvesting the food consumption for human. At the same time, we can use the second step, we can use the bacteriophage as a biocontrol agent as a food preservative or post-harvest pathogen control. Sometimes we can use the bacteriophage therapy to prevent the production of salmonella or Listeria monocytogen in meat stock or agriculture stock. Sometimes we can use the bacteriophage in the biosanitation protocol in some factory and industry. Some factory and industry has important biofilm production from bacteria. This can be found in the steel or the surface in the factory or industry. So we can use bacteriophage to control such bacterial agent that can grow in the surface inside the industry or factory. Finally, we can use the bacteriophage for food preservation for human consumption. So this is the term means farm to fork, or means from pre-harvest control to human consumption control. So we speak more about bacteriophage, but where can we find the bacteriophage? Bacteriophage can found in anywhere in nature. So we can isolate bacteriophage from evening inside in front of you now from the table, from soil, from water, from plant material, we can find the bacteriophage. This is like a technique of isolation of bacteriophage. So we always say the seawater is an important source for the isolation of bacteriophage for agriculture and veterinary and human consumption. So sometimes we take the environmental sample and make some filtration to get the bacteriophage, and after that make incubation of the bacteriophage with the host bacteria needed for a treatment. So for example, if we have a Pseudomonas aeruginosa or a Listeria or Salmonella, and we need to make isolation for a specific bacteriophage to kill this one. So we take the sample of this bacteriophage and incubate with the host strain like Salmonella or Pseudomonas aeruginosa and make enrichment of the bacteriophage to produce billions of bacteriophage. After that, we make culturing of this mixed culture of bacteriophage and the bacterial cell to produce plaque like that of antibiotic disc in the plate agar or agar tissue. So after that, we make serial dilution to find the specific plaque that make specific killing for this strain. After we found this plaque, which is specific for killing this strain of Pseudomonas aeruginosa or Salmonella, we can produce more stock to this bacteria. This is like this one of our experiment inside the University of Agriculture in Japan. This is the step of agriculture of enrichment, or propagation of the host. This tube contains a mix between the bacteriophage we isolated from the soil and the bacterial strain isolated from the same site. So we make enrichment here, and after that, put them in the double layer agar method. This method is specific for bacteriophage isolation to produce a single plate like this one. Do you see this bacterial culture? And we can see a million of small plaque. This is a million of small plaque here like this one. This is each one plaque of this contain a million of bacteriophage. So we should isolate one plaque of this one until we make a specific strain of bacteriophage to kill a specific strain of Pseudomonas aeruginosa or Salmonella or host strain. Sometimes this bacteriophage can produce two plaque. One of this plaque can make complete lysis of bacteria like this one, and the other bacteriophage can make incomplete lysis of bacteria and produce a turbid plaque. So there is a small growth of bacteria inside the plaque. So when I wanted to use or choose bacteriophage to treat one bacteria, we mainly prefer to use that one bacteriophage that produce clear plaque, because this turbid plaque or this bacteriophage not completely destroy our bacterial strain. These are some products used in the biocontrol for food safety. This product, SalmoFresh, List Chilled, and EcoShield, each one named by the bacterial strain. For example, the SalmoFresh means this bacteriophage can treat salmonella, and the List Chilled means that bacteriophage can effective against Listeria monocytogenes, and the EcoShield means that bacteriophage can specific for E. coli. These are most of bacteriophage products. They are found in the commercial market. So they are now used or produced by Interlytics company, and they are very famous now in food preservation and factory or industry of food safety. So this is I nearly finish my presentation, and this is some of our paper or our publication to treat or to explore some bacteriophage application in medicine. And if someone want to interested to read some of this publication, he will find more information than this presentation. And I would be happy to, if someone need more question or more contact, this is my email contact and the Google Scholar and the ResearchGate. If someone has question or any other thing, I will be happy to assist you. So thank you so much for listening, and thank you for this invitation.
Okay, thank you, Mr. Ahmed, for delivering us this interesting topic tonight. And I believe most of the participants here cannot wait for the discussion session. Therefore, let us start the discussion session. So the discussion session will be limited with 3 questions. So please, to all participants who have question, raise your hand and introduce yourself to ask question. Please, any question? Okay, maybe before we start with our participant, I have a little question related to your topic because it's quite interesting, bacteriophage. And is there any side effect or negative effect in implementation of bacteriophage in large scale, especially because we are the agro-industrial technology, so we talk about industry, and most of industry have some kind of waste side product, and it maybe can be implemented to clean up the pathogen in the waste of industry, the liquid waste of industry. So from your experience or your expertise, is there any negative side effect, Mr. Ahmed?
Oh, yes, thank you so much for this question. This is very important question. This question, someone asked me this question in my PhD presentation in Japan. So thank you for this question. Until now, there is no a specific disadvantage or side effect for the using of bacteriophage. Only one study reports that the using of bacteriophage initiate or produce some immune response in human body or animal body. So this immune response sometimes can make some negative effect for the human or animal tissue. But this only one study. So until now, the only one side effect for the bacteriophage is the live microorganism of bacteriophage. I mean that bacteriophage not like antibiotic. Antibiotic just a protein or drug, but bacteriophage is a live microorganism like virus, like bacteria. So until now, we can't control if we infect, for example, those with a specific concentration after injection inside human or animal body. This dose may be replicate and increase in the concentration because of replication of bacteriophage. So this is the only side effect we facing now for bacteriophage production as a large scale. We can't make a standard experiment or standard result to control the dose of bacteriophage. But as a side effect, there is no specific side effect for the using of bacteriophage. Thank you.
Thank you for the answer, Mr. Ahmed. Okay, so I will let all the participants. Any question? Okay, Mr. Miftahul Khairan, please.
Okay, thank you very much. Very interesting. Talking about the bacteriophage. With the Okay. Let me ask about application of a bacteriophage in as a biopreservation in food industry. So, how can the bacteriophage withstand with the food processing? We know that food processing sometimes use the high temperature or low pH or something like that. So, how can that bacteriophage withstand with that condition? Thank you.
Okay. Okay. This is a good question also. Thank you so much. Um, Before using one of bacteriophage for either treatment or biopreservation, we should make some experiment in vitro in lab to check the availability of this bacteriophage to adapt it in this new environment. I mean that there is many experiment we can test the pH where bacteriophage can live, acidic or alkaline, which temperature the bacteriophage. Some bacteriophage can work in a high temperature and some bacteriophage can work in a low temperature. So before using or choosing the specific bacteriophage, we should test this bacteriophage against the different environment when used in a factory or industry or something like that. So if this bacteriophage not work in high temperature, so we should search for another one that is able to work in a high temperature. So and the opposite for the cold temperature, and the same for the pH. We know we should choose the bacteriophage to be optimum for the pH and the temperature inside the factory or industry.
Okay, so basically we need to search the correct bacteriophage for the specific product,
Yes.
right? Oh,
Yes.
okay. Thank you very much.
Okay, thank you.
Okay, so just to clear up, Mr. Ahmed, so every bacteriophage has its character, yeah?
Yes.
Okay, thank you.
Yes.
Any other question for the participant? I think it is quite interesting because I don't know for in Jember, for example, I don't see a lot of industry has implement using bacteriophage, but if we know about this advantage of bacteriophage, maybe most of industry can reconsider about that. Yeah, instead of antibiotics or any other physical treatment using machine, and it will be costly, yeah, for industry. And I think it's a great idea.
Of course. Thank you so much.
Okay, do we have any other question from participant? No? Okay, I think that is all for our first session. Thank you very much, Mr. Ahmed, for your lesson. It is quite helpful and useful for us, of course, in the future.
Oh, thank you so much for your introducing myself, and thank you so much for Dr. Muftah Khair for this invitation. I am happy to be with you. Thank you.
Thank you.
Assalamu alaikum.
Waalaikumsalam warahmatullahi.
Salam.
Thank you very much, Dr. Ahmed.
Okay, okay, so we can step into our next guest lecture, ya, Mr. Firoz. Mr. Firoz is here. Okay, so before we start, maybe I can read for the curriculum vitae of Mr. Firoz. Okay, so his name is Dr. MD Firoz Mortuza. Okay, his first education of bachelor degree is a Bachelor of Science, Biochemistry and Molecular Biology. And then for the master degree, he went to University of Dhaka, Bangladesh, Master of Science, Biochemistry and Molecular Biology also. And the last one, the PhD program. The last one for the PhD program, he went to the United Graduate School of Agriculture Science, Tokyo University of Agriculture and Technology, Tokyo, Japan, for agriculture science. So, sorry. Okay, so for today's session, we can start to Mr. Firoz. Is Mr. Firoz still here?
Assalamualaikum.
Waalaikumsalam, Mr. Firoz. Good afternoon.
Good afternoon. Yeah, how are you?
Thank you, thank you. I'm fine. How about you, Mr. Firoz?
Yeah, can I start?
Oh yeah. Sure.
Okay. Okay. Can I share my window?
Yes, please.
Okay. Can you see my screen?
Yes.
Okay. Please let me know if there is an internet connection is not very good here. Okay. So, Assalamualaikum and welcome to my presentation. The title of my presentation is Application of Irradiation for Agriculture and Food Product. I am Dr. Mohammad Firoz Mortuza. I'm working as Principal Scientific Officer in Gamma Source Division, Institute of Food and Radiation Biology, Bangladesh Atomic Energy Commission. The contents of my presentation, first of all, I'll talk about the objectives, then basic information of radiation, and then mode of action of radiation and biological effect, application of irradiation in food processing, and activities of Gamma Source Division. Objective. Main objective is to provide some useful information about radiation and to improve understanding of radiation, what it is and how it interacts, and to share and discuss about the effects of ionizing radiation, especially gamma radiation, and to inform about the applications of cobalt-60 gamma irradiator, especially in agriculture and food. Information of radiation. The Marie Curie said that nothing in life is to be feared. It is only to be understood. And now it is the time to understand more that we may fear less. So it is very important to understand about radiation because we have a kind of a phobia about radiation. So it is better to
Yeah, I think there is some trouble with the connection.
can you see my screen?
Yes, we can see.
Okay. Assalamu alaikum, and extremely sorry for such a awkward situation. Yeah, my laptop somehow became black. Okay, now Alhamdulillah it's on. So now, as I am saying, so we should understand about radiation. So these are some of the basic information of radiation. So there are actually two types of radiation depending on their ability to cause ionization. One is called ionizing radiation, and the other is non-ionizing radiation. So as you can see, suppose there is ultraviolet ray, visible infrared, microwaves, radio frequencies. These are all non-ionizing radiation. They cannot cause any ionization. On the other hand, there are X-rays and gamma rays which can cause ionization. So they are called ionizing radiation. And there are also two types of radiation depending on their type, like particle type and wave type. Particle type are alpha particle, beta particle, neutron beams, and proton beams. These are particle type. And there are electromagnetic waves like X-ray and gamma ray. So these are called wave type. So I made this animation to make a better understanding of how gamma ray interacts with matter. Suppose there is a gamma ray incoming gamma ray, and when it acts with the electron, how it works. This is suppose the gamma ray, and so it hits the electron of an electron shell, and the electron ejected from the shell, and this electron has the energy of the gamma. So now this electron, it will act like a beta electron, and afterwards it will also ionize. So this atom will become positive charge, and afterwards this electron will act like a beta. So there is another way gamma ray reacts. This is called Compton effect. In a similar way, it ejects one electron, and also it really some of the energy is still there in the gamma. So this gamma will react with another electron, and by this way, it will continue reacting. And another way is when the energy of gamma is very high, then it reacts with the nucleus and it produces electron and positron. This positron, again, when it collides with electron, then it produces two photons. This is called pair production. So these are the effect of gamma. Afterwards, another is beta. So how beta works? Beta, we know beta is negative charge. So when it passes through electron shell, so an electron will be ejected from the orbital, and this way beta will react. And alpha, we know alpha has positive charge. So when it passes with atom, so it will attract the electron out of the shell, and the atom will become positive charge, and there will be some ejected electron that will act like a beta. So this is how gamma, alpha, beta, they reacts with the atom. So by this way, in the table they have charges like +2 charge, and beta has a -1 charge, and gamma it doesn't have any charge. So the range in air, alpha can travel very little distance, beta little bit more, but gamma can travel long distance if it is not interacted with any electron. And the problem is alpha. Alpha, if it is ingested or inhaled, then it creates lots of problem. And beta, superficial skin injury and whole body injury at a very high dose. If the dose is very high, then gamma is risky. So alpha, beta, and gamma. Alpha can be stopped within the skin, and beta can penetrate the skin, and it can react in the tissue, and gamma can penetrate the skin, tissue, and bone and other organs. So this is how we can stop. Alpha can be stopped with a very thin paper, and beta can be stopped with thin aluminum, and gamma rays can be stopped with thick lead shield. And neutron, it needs water. Water is very good shield for neutron. Units of radiation measurement, radioactivity is measured in becquerel. 1 becquerel equal to 1 disintegration per second. It means that if one atom is broken down, one radioactive atom is broken down in 1 second, then it is called 1 becquerel. And another is absorbed dose. It is measured in gray. So suppose some person is getting radiation. So in that case, we will say how much gray that person absorbed. Like this. And equivalent dose is measured in sievert. Effective dose is measured in sievert. These two I will explain later. So here, radioactive material is measured in becquerel. So from here, radiation will come, and suppose someone is getting that radiation, absorbing that radiation. So amount of energy absorbed by a substance of unit mass that received radiation. It is called absorbed dose. Absorbed dose is the amount of energy absorbed by a substance. And suppose total energy divided by the weight of that person, the mass of that person. So by this way, you can calculate the rate. Now, there is depending in effects. There is difference in effect depending on types of radiation, like equivalent dose, and also there is difference in sensitivity among organs. So we calculate for effective dose. This will make it a little bit easier to understand. Suppose equivalent dose is radiation weighing factor into absorbed dose. Suppose one person absorbed 1 gray dose, and if it is gamma, then we will multiply with 1. But if it is alpha, then we have to multiply with 20. So that means 20 sievert. So it is very dangerous for alpha and heavy ions. And similar way, that effective dose is measured with tissue weighing factor into equivalent dose. The equivalent dose that we are getting, then we will multiply with different weighing factor. Like here you can see that highest red bone marrow, colon, lungs, stomach, these are highest weighing factor, and bone and skin is the lowest. So by this way we can calculate how much effective dose we are getting. It is also measured in sievert, remember. So here you can see that the cells that have active cell division, these are highly sensitive, like bone marrow, lymphatic system. These are very sensitive to radiation. But nerve cell or skin or this that has lower cell division, so they are less sensitive to radiation. Now, why is radiation? So actually, we have radiation inside our body. The most important natural radionuclides that are found in the body are uranium, thorium, polonium, lead, potassium, and carbon, also carbon-14. So we have many radionuclides inside our body. So if we count only about potassium, there is 4,375 becquerel. That means in each second, 4,375 radioactive potassium is broken down in our body. It releases rays, and so we have this amount of radiation also inside our body. And yearly dose, we get 1.3 into 10 to the power minus 4 gray from potassium only. So we get radiation from the, as I said, inside internal radionuclides, we get cosmogenic radiation, we get cosmic radiation, and we inhale. There is radon that we get from the earth, earth. Radon is produced. So we inhale. So we get radiation from our environment, and totally, a human being absorbs about 3 millisievert dose per year from nature. This is we get from the environment and from our body. Now, mode of action of radiation and biological effects. Okay. So, as I said that gamma ray and X-ray directly it ejects electron, and afterwards water atom it reacts with water, and water is broken down to hydroxyl ion, hydrogen ion, and there are also free radicals. These then indirectly react with adjacent biomolecule. So these are very active free radicals or ions. So it will react with the DNA, RNA, protein, etc. So because the short life of simple free radicals, the short life is very short, 10⁻⁻10 seconds. Only those formed in water column of 2 to 3 nanometer around the DNA are able to participate in indirect effect. That means that suppose we get radiation and there is water molecule, and water molecule is broken down because we have a very high amount of water in our body in every cell. So water molecule will be broken down, and that free radicals will react with DNA. Now, why we are basically more concerned about DNA, as you know that we have like 23 pairs of chromosome, but we only one single set of chromosomes. But if we have, because we have protein and RNA and other things, if they are destroyed or denatured, then it can be reproduced from the DNA. So, but if DNA is, there is a mutation and problem in the DNA, then we suffer a lot. That's why we are mainly concerned about DNA. Now, how types of DNA lesion, how different types of DNA lesion are produced by radiation. And here you can see that 1 gray of dose of gamma radiation can produce base damage and deoxyribose damage and single stranded break and double stranded break. These double stranded breaks are very lethal. Like if it breaks both the strand, then the DNA is separated, so it causes lots of problem. But we have DNA repair system, but still it's a big problem. So 1 gray here you can see that 1 gray of radiation can produce many problems. Okay. Now, fortunately, we have cell has complex signal transduction, cell cycle checkpoints, and repair and pathways to respond to DNA damage. We have many repair system inside our body, which is very helpful for us. So suppose there is DNA damage, and if the cell will try to repair, if the repair is incomplete, the mutation, and then there is possibility of causing cancer. And if repair fails, then cell death occurs, and due to cell death, there is a possibility of causing acute effects and fatal effect. If the dose is very high, then the effect is very high. But if repair succeed, then there is no hazard. This is also a similar thing. The normal cell amount of radiation is low, and there is some damage. Then even if there is dead cell 1 or 2, but the cells are repaired, so there will be no effect because a tissue has lots of cells. If the radiation is a little bit high, then there will be many cell death. Then temporary dysfunction will occur. But if it is very high, the dose is very high, and there are lots of cell death, then loss of function and morphological defect will occur. Now, in human body, there is antioxidant process to neutralize free radicals and very efficient DNA repair system to repair DNA damage. So here, this is one research from nucleic acid research. It is published in the nucleic acid research. So here you can see that that these are the pre-irradiated cell, and these are called foci. These are called foci. This represents double strand break. Just to understand that these dots are double stranded break. So here you can see 100 milligray dose. There are 2 or 3 dots. So there are 2, 3, 4 double strand breaks occur in the cell. If the dose is high, 500 milligray, then about 10 double strand breaks. If it is 1000 milligray, that means 1 gray, then here about 20 double strand breaks. These are different types of marker. They do the same thing, but different markers. So here you can see that number of foci per cell with the increasing dose, the number of foci increases. That means number of double strand break increases. Now here, the repair system, you can see that with time, the repair system works very well. So, so here you can see that with 2 hours, 4 hours, 6 hours, and the cell repairs, and it goes down. Suppose after 8 hours, if we have suppose 20 double strand break, then it goes down to 4, and with time it even goes to 0. It even goes to 0. So this is how it works. Our cell, they have very good DNA repair mechanism. So the effect of radiation or other effect also, they try to repair that damage. So what things to compare? What causes cancer? We think that radiation, and with radiation there is mutation and cancer. Whenever we hear about radiation, we become frightened and we think that this is the only thing that causes cancer. But here you can see that gamma radiation, as I said, that double strand breaks are very hazardous or lethal. So 1 gray produces 20 to 40 double stranded break. But cellular metabolism per cell per day. Every day we are working, we are eating, we are running. So we have cellular metabolism to produce energy. So per cell per day, there is this kind of damages occur. Double strand breaks, single strand breaks, and DNA based damage, these occurs in our cell. But our efficient, alhamdulillah, such a very good repairing mechanism that makes our cell healthy again. So 3% of metabolized oxygen is converted to free radicals during cellular metabolism. About 70,000 relation is produced per cell per day due to cellular metabolism. This results in an average of about 1 mutation per cell per day. But whereas background radiation, as I showed you, that we get 3 millisievert per year. So here, if we compare 1 millisievert produces 10 to the power minus 7 mutation per cell per day, which is very, very low compared to the cellular metabolism. So we should think about it. Now, there are 3 basic rules about radiation protection. Whenever there is radiation, we should be careful. We should not be frightened unnecessarily, but we should take proper care. We should not get dose unnecessarily. So we should be very careful about this. So less time, we should stand less time. We should maintain distance as much as possible, and we should take shielding. Now let's talk about application of irradiation in food processing. So So traditional, there are different types of food processing are there, like traditional food processing and radiation food processing. So traditional food processing, like there are methods of fermentation, sun drying, smoking, salting, whereas radiation food processing, there is gamma radiation, electron beams, X-rays. You can see that efficiency, these are labor intensive and time consuming, whereas for radiation food processing is highly efficient and quick process. But the cost is low in traditional food processing. So it is a positive side of, from this perspective, but in case of radiation food processing, it is very high initial cost and require specialized equipment. Quality, in case of traditional, it is inconsistent and varies with condition, whereas radiation is very consistent and controlled environment. So health impacts, potential health risk due to contamination is there, but in case of radiation, it eliminates pathogen very efficiently, improve food safety. And by this way, so there are many positive sides of radiation food processing. So and the World Health Organization, WHO, FAO, and IAEA, they had very high level of research, and they concluded that up to 10 kilogram radiation dose for food, it's completely safe, and then there is very little change in the food, nutrition, and other things. So now application of irradiation. Gamma irradiator are equipped with high activity radioactive source of cobalt-60 or cesium-137, which are used for different purposes such as sterilization or decontamination. So there are many applications of gamma radiation. You can see for medical, pharmaceutical, tissue based biological radiotherapy, cosmetics, toiletries, research and development. And there you can see that in case of radiotherapy and R&D applications also we can use gamma radiation, many types of radiation. One process and multiple use. We can use for quarantine and pathogen reduction and shelf-life extension, insect disinfestation, and sprout inhibition also. So food irradiation is a technology which applies ionizing radiation to food for improving the safety of food and extending the shelf life of foods by reducing or eliminating microorganisms and insects. So it is used over 60 countries and more than 500,000 metric tons of food. Now it is even more. So day by day it is increasing food irradiation in many countries. So it's good. And there are many types of sources, basically gamma rays, and now X-ray and electron beams are also used for food processing. As I said, that one process, multiple use, it can use for prevention of foodborne illness, to eliminate organisms, and for preservation, to destroy inactive organisms that cause spoilage and decomposition. It can also use to control insects for phytosanitary purpose to destroy insects and delay sprouting and ripening to inhibit sprouting and delay ripening of fruit to increase longevity. And sometimes it can also use for a high dose sterilization to completely kill the bacteria and other organisms. So depending on purpose, there are different type of doses that we use for irradiation. As you can see, low dose irradiation, it is called radicidation, and like inhibit sprouting, potato, onion, yam, garlics. So for this, we use a very low dose, 0.06 to 0.2 kilogram for delay in ripening. Like this, these are for low dose irradiation. And there is medium dose irradiation that is called radiolization. So we can say this for extend shelf life of raw and fresh fish, extend shelf life of refrigerated and frozen meat, reduce risk of pathogenic and spoilage microorganism to reduce, not completely destroy, to reduce. So increase juice yield, reduction in cooking time of dried vegetables. This dose varies from 1 to 10. So this is called deodorization. And for high dose, like deodorization for sterilization of spice and dried vegetable seasonings. This is although it is 30 kilogram max, but we can use even 10 or less. Uh, so now you can see sterilization of packaging 25 kilogram and sterilization of foods for NASA and hospital. This is for high dose we can use. So these are the application of irradiation in food processing. Now let's talk about my division. I work in gamma source division. It was the gamma irradiator was established in 1979. The initial activity was 50 kCi. Then in year 2000, we brought a new source, which is 50 kCi from India. Then again, we extended the source to 65 kCi in 2009. And in 2014, it was extended to 90 kCi. Now we use different type of dosimeter to measure the radiation dose, how much radiation was absorbed in the product. So we place the different type of dosimeters. Here you can see Fricke, amber perspex, red perspex, seric ceros, and galvanic and alanine dosimeters. There are different types of dosimeters that we use, and they have different purposes. The dose range is different. Fricke is for low dose, and these are for high dose. Galvanic is also for low dose, 1 to 1000 gray. Others are for high dose irradiation. So these are some of the pictures of the dosimeters, the amber, perspex, seric ceros, red, Fricke, alanine, and galvanic. These are the machines that we use to measure the dose. Tensiometer, UV spectrophotometer, and dose reader. We prepare Fricke dosimeter by ourselves, and after preparation, then we calibrate in secondary standard dosimetry lab. It has a teletherapy unit, and there is a farmer type ionization chamber. So we use this facility to calibrate our prepared Fricke. The dosimeter that we prepare. So after preparation, we perform the calibration. If it is okay, then we use it. So we had a dosimetry inter-comparison test in about 13 countries participated. I was one of the chief scientific investigator of that dosimetry inter-comparison. So the reference dosimetry certificate was provided by Ariel France under IACRP. In that test, so there was target dose of 100, 400, and 1000 gray. The Ariel and France, they determined our dose about 101 and 396 for 400 and 1010 for 1000. So there was only about 1% variation was there. So according to the expert, it was a very good result. So activities of gamma source division. So we use gamma radiator for research and development at free of cost and commercial purpose to earn revenue. And so all the divisions of our institute, Bangladesh Atomic Energy Commission, and also other research organizations all over the country, they send their sample to us to irradiate and give them service, and we do it at free of cost. And other food industry and medical and pharmaceutical company come to us, so we earn revenue from there. R&D services, these are different types of product like food and agriculture samples. Here you can see various types of food. We irradiate seeds of rice, pulse, gladiolas, in vitro plant tissue, fish, jute, chickpea, vegetables, seed. We irradiate and medical samples also. There are different types of medical samples, pharmaceutical samples, insects, and other samples also. We irradiate for R&D purpose, research and development. More the organization came to us in last few years, and we irradiated more than 2,000 food samples and about more than 18,000 medical polymer and other samples. These are for commercial services. There are different types of commercial services like medical goods, pharmaceutical raw material, pharmaceutical finished product, and pharmaceutical packaging products and food products. Here you can see also we irradiate spices, pet food, and spirulina seasoning and peanut. These we irradiate, and uh, more than 60 countries, 60 companies came to us, and we irradiated more than 386 tons of food product and more than 37,000 CFT of medical and pharmaceutical product in commercial service. So these are just some of the pictures. Spirulina, turmeric, mushroom, ballistic, here. Conclusion, radiation is all around us and it has many beneficial applications. The issue of radiation exposure is not a matter of safety only, it is also a matter of benefit compared to risk. So there is a concept called concept of LRL, as low as reasonably achievable. So we need to be cautious and careful about radiation, but we should not be frightened unnecessarily. There is one small video. So I think it will be helpful to how much radiation we get every day. So living within 50 miles, airport is 0.1, eating a banana, 0.1 microsievert. Dental X-ray, 5 microsievert. New York to Los Angeles flight, 40 microsievert. Yearly release limit, 250 microsievert. If we do CT scan, then 1000 microsievert. Average yearly dose from natural source, 3.1 millisievert. Smoking 1.5 x, 13 millisievert. This is the maximum dose, and dose causes symptoms, 40,000, 400,000. And that's it. So thank you very much. In Bangla, we say apne connect dhonnobad. Terima kasih. And I am extremely sorry again.
Okay. Thank you, Mr. Firoz, for your lesson material. Okay, next one, we step into our discussion session, which I reckon have been waited for all the participants. Okay, so for this discussion session with the same rules as before, any participant who have question related to this material, please raise your hand. Okay, any question? Okay, maybe, okay, yeah. Please, Mr. Miftahul.
Okay, thank you very much, Dr. Firoz, for your interesting presentation. About the radiation, actually, I didn't see many facilities in at least in my city, for the irradiation facilities to create the radiation. Maybe I can see in a hospital or any medical facilities, but in food industry, I didn't many facilities for that. So, what do you think about the if industry of if the food industry has their own facility to make irradiation, is it better uh or is it better for them to send the sample to the for instance your facility to treat the sample with the irradiation like a gamma or x-ray, something like that? Thank you.
Okay, suppose there are actually many things that are involved in case of radiation and radioactive sources or gamma and X-ray like this. There is like about environmental protection, physical protection, the protection of worker. So these are kind of specialized machines or specialized facilities. So uh, so actually, if the government and private companies they can manage, it can be anywhere actually. But there are many prerequisite. There are many things that needs to be handled properly. So these are something that actually a matter of concern. But there are many countries, even Indonesia, there are many radioactive sources now that are used in agriculture irradiation. In China, they have many electron beam and gamma irradiation. Uh, but there are some issues because radiation it needs to be handled properly. So there should be proper management, proper care, proper safety issues should be addressed. If everything is okay, if the regulatory authority, if they permit, then the food industries they can even set up their own irradiator. It can be E-beam or X-ray or gamma, but these things should be met up properly.
Okay, thank you. So how long the treatment should be taken for the food preservation maybe? How long the irradiation
Or
process for the food?
suppose for food preservation, the dose if normally suppose for phytosanitary irradiation, we will need a dose of about 400 gray only, 400 gray. For a facility like us, it will take only few minutes. But if it is E-beam, it will be performed in seconds. Even in case of E-beam, 25 kilogram dose can take only 30 seconds to 1 minute. And this is very fast and very high energy beam. So they can process product very quickly, but there is also some limitations as maybe I am saying sorry because of the interruption that occurred. Suddenly it happened or anyway. So as I showed that when electron interacts because it has charge, so it loses its energy very quickly. So it is high. It is called linear energy transfer (LET). So it is a high LET object kind of. Like this alpha and electron, but gamma it can penetrate a long way without interaction. So it is called low LET. and there are some issues, like in some cases gamma is good, in some cases E-beam is good. And if you remember, I showed that gamma and X-ray, they are actually same. They are both of them are electromagnetic wave type, but gamma is produced in the nucleus, and X-ray is produced in the electron shell, from the electron shell. This is the different. If gamma and X-ray energy is same, then it will react with material in the similar fashion. Otherwise, electron beam and gamma, they have different purposes and different way of use. For same thing, we cannot use both. So there are both pros and cons of these machines. And it takes very little time actually.
Okay, thank you very much for your explanation.
Okay, thank you for the first question. Is there any question again from the participant? Okay, if there's
Want
no
to
question,
ask
yeah,
a question for the speaker, Dr. Ahmed. So my name is Liduhayu.
Dr. Firos,
Oh,
sorry.
Dr. Firoz, sorry.
Mhm.
My name is Liduhayu. I want to ask a question about the industrial scale of the irradiation. So it's the very interesting presentation from Dr. Firoz, but I want to ask you something about the application of irradiation in industrial scale operations. Is this anything you know about the scale in industrial, the larger scale of irradiation for export industry? So we in Indonesia can apply this irradiation technique in our industries like that.
Okay,
I think
thank you. Mhm.
actually you have like BRIN.
Yes.
Brin, yes.
Yes. But previously maybe Batan. So they you have radioactive sources. You have E-beam in your country, and Indonesia, as far I know, they there are lots of industrial application is in Indonesia also. And I work in IAEA CRP and in another project. So there are many Indonesian scientists also. I met with them. They use E-beam and gamma irradiator also for industrial application, a large scale industrial application also.
Okay. For standard in export quality, we have some regulations from the government and from BRIN also. So in the application of irradiation, we can use this for export quality. Is that so, Dr. Firoz?
Again, please, the last part, what did you say?
So you can think we can use the irradiation thing for our industry to maintain our export quality. Is that so, Dr. Firoz?
Yeah, of course. Actually, uh, as per my understanding and knowledge, it is expanding day by day. Not only in Bangladesh, also many countries, Indonesia, also China, Vietnam. They are expanding their irradiation facility, and for wherever there is food and vegetables and even pharmaceutical and medical products are there. They try to go for exportation. So these irradiation sources help for export very much. So I think that uh, it's time actually to go for irradiation for more export for our product. Yeah.
Thank you so much for the insight. I hope our students here will be future food technologists that we can use irradiation for more future export quality of our agri industry. Thank you very much for the answer, Dr. Firoz.
And another thing also that suppose the quality of food, like if we want to export mango, suppose in Australia, the two the for phytosanitary purpose, it should be either it should be irradiated or it should be boiled 50 degrees Celsius in boiled water. So you know that if we boil a mango, then the taste will not be there. So it is better to irradiate. Like this, many product, if we use a traditional or other method, then the food quality it degrades. But radiation is very promising in those cases. So case by case or product by product, we can choose which one we should select. But of course, radiation is such a good thing in these cases. We can go for radiation in for many, yes.
Thank you so much for the insight.
Okay, thank you for the question. Okay, because this session is contain 3 questions, yeah. So I will add the last question, I think. Okay, it's related to the investment, Sir Mr. Firoz, because most of Indonesian industry, like almost 80% of Indonesian's industry is from mid to below level of industry. So how about the cost if we use the irradiation? Um, do you recommend, or is it effective for most of the industry, especially for the mid to below level, or is there any other solution that may be cheaper for our mid to low industry? Thank you.
Yeah, actually, the initial cost of establishing irradiation facility is high. Initial cost because of preparing the building. It needs very thick walls, and there are some regulation due to some regulation. So initial cost of making the building and this thing is little bit high. But after the machine cost and afterwards the replenishment cost of sources, these are not very high. Suppose establishing a e-beam facility, maybe it will cost about $3 million, suppose $3 million, suppose. Within few years, if it runs 24/7, and it can be run 24/7 and most suppose 360 days or 350 days per year, the turnover will be very good. Within few years, it will be uh, in the beneficial side. But the thing is that there should be enough product. Like if company, if they don't have enough product, if they set up this kind of machine, then it will not be used properly. But multiple companies, if they can manage, it will be good. I mean, as much as we can use radiation, then it will be it will be very cost effective. It will be very beneficial for us. And I think that actually uh, 2 million, 3 million is not a very high amount of money, but within few years, you can get the money back on the export. Yes.
Okay, thank you. So actually for irradiation, it's a good investment, yeah, because it's not a long-term return, so it could be a short-term return comparing for the benefit itself, yeah, and its effectivity. Okay, thank you, Mr. Firoz, for the lesson and the explanation. Okay, so I think that is all for tonight discussion. Maybe next time we wish to have more time, yeah, next time to discuss more related to these two topics, which is very interesting. And let's step into the last agenda, which is closing and announcement. Therefore, to close our guest lecture today, let us say Hamdalah together. Alhamdulillahirabbil alamin.
Alhamdulillah.
Amin.
Okay, I believe the lesson we have learned from our international guest lecture speaker tonight will be very useful to our knowledge and to support our competence as agro-industrialists in the future. Yeah. Please let me thank one more time to our international guest lecture today, Mr. Ahmed and Mr. Firoz. Thank you very much for sparing your time for us, for sharing related to your expertise.
Thank you. And from my part, again, I'm sorry for wasting your time. I am very, very sorry. Please don't mind. Yeah.
Yeah, thank you. It's okay, Mr.
May Allah,
We can understand.
may Allah forgive us all. Yeah.
Yeah, we
And
are
for
very
me, thank you so much for me also. Thank you. And thank you for meeting Feroz Sam. So
Ahmad
long time
Sahib.
I didn't see him. So I should thank you for this invitation to see us.
Yeah. And
So
from my
one
part,
of
yes,
the purpose of this meeting is to meet you. So
reunite.
Meet each other.
yes, maybe next meeting we will gather more alumni from QAT.
Hopefully so.
it is nice
Assalamu
to
alaikum.
see you all.
Uh,
Yeah.
okay, I think we are very grateful for having both of you, sir. And thank you also for all participants who is attending this guest lecture tonight. And however, before we close the event, let us take a picture together as a documentation. So please turn on your camera. I will take a picture. Your camera, yeah. Okay.
Enggak.
Okay, the first page, yeah. We have a few page, I think, here because there are a lot of participant. Okay, I will count to 3. Okay. 1, 2, 3. Okay, hold on. Okay, so next one, next page. Okay, next page. Ready? 1, 2, 3. Okay. Okay, third page. There's no video. Okay. Sorry, I think there is a problem here. Okay. Okay, finished for the documentation. I would like to apologize also as a moderator if I made some mistake here and there during the guest lecture. And I'll see you in another guest lecture webinar. Also for student, don't forget, next week we will have another webinar, national webinar with Indonesia University, Udayana University. So please attend our another webinar. Jadi minggu depan akan ada ini ya, webinar lagi ya bersama dosen dari Udayana. Jadi kita bisa sharing-sharing lagi. I think that is all for Mr. Firoz, Mr. Ahmed, and all participants. We had a great time together. See you in another guest lecture webinar. Wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Oh.
Waalaikumsalam warahmatullahi.
Thank you so much, Mr. Ahmed and Mr. Firoz.
Thank you.
Thank you.